We know if we can get the group together. All right, good morning. Welcome to Grand Rounds this morning. What a pleasure it is to introduce Dr. David Perlmutter, who will give this morning the inaugural David Perlmutter Lecture in Pediatrics. This lectureship was established by David's colleagues when David was here as Chair of Pediatrics. And I'm honored to be able to host David for the very first David Perlmutter Lecture in Pediatrics. Dr. Perlmutter is the Spencer T. and Ian W. Owen Distinguished Professor, Executive Vice Chancellor for Medical Affairs, and the George and Carol Bauer Endowed Dean of the Washington University School of Medicine. And as you know, he is also my predecessor as chair of the Department of Pediatrics. Dr. Perlmutter is an internationally recognized leader in studies of alpha-1 antitrypsin deficiency, a genetic disorder in which accumulations of a misfolded protein can cause severe liver damage. His work has led to advances in an understanding of how cells dispose of misfolded proteins that are toxic and can cause cellular damage and dysfunction. He also has developed a pipeline of drugs, including one that is currently being evaluated in phase 2 clinical trials for treatment of those who have alpha-1 antitrypsin deficiency. And what's so exciting about this work is it has the potential to really eliminate the need of liver transplant to treat this condition, which is currently the only therapeutic option for those with progressive liver damage due to this condition. Even more exciting, because these drugs enhance normal processes of protein turnover, they have the potential to prevent cognitive decline and other degenerative diseases of aging. And I know what you're thinking. David was going to tell me a little bit about these drugs, which may help with some of these premature processes, at least premature in my opinion. Now, a bit more about Dr. Perlmutter. He received his MD degree from St. Louis University School of Medicine, completed pediatrics training at the Children's Hospital of Philadelphia, and underwent his GI fellowship and research training at Boston Children's Hospital. He held faculty positions at Harvard Medical School and Washington University before serving as chair of the Department of Pediatrics here at UPMC Children's Hospital from 2001 to 2015. He's been in his current roles at Washington University since that time. Dr. Perlmutter is a member of the National Academy of Medicine and also the American Academy of Arts and Sciences. He is the past president of the Society for Pediatric Research and a past member of the Advisory Council of the National Institute of Digestive Diabetes and Digestive and Kidney Diseases. He's been honored with many awards for his research, including the E. Johnson Award for Research in Pediatrics from the American Pediatric Society. Now, David's going to give 2 talks today, his Grand Rounds presentation this morning, and then another special talk on his science in more detail that will be at noon today in the Plaza Building. It's 300 in the Plaza Building at noon. But this morning he's going to talk about his studies of alpha-1 antitrypsin deficiency, and the title of his inaugural Perlmutter lecture is Antitrypsin Deficiency, From Rare Genetic Disease in Childhood to How We Age. Please join me in welcoming David Perlmutter back to UPMC Children's Hospital.
Thanks so much, Terry, and thanks for doing this. I want to thank Children's Hospital Pittsburgh Foundation, Children's Hospital, and UPMC, which were all part of the, and I'm, Terry, forgetting the year, but it must have been like 2013, 2014, and this $10 million and was dedicated to research. It was given to the Department of Pediatrics. And Rachel will know the whole story. This was a very clever way of getting $10 million to the Department of Pediatrics and dedicated to research in the department. And UPMC agreed to it at the time. And so I think we also have to thank Jeffrey Romoff, who signed off on the and I have been thinking a lot about him lately. And thinking a lot about him lately in terms of some of the goings on at Washington University School of Medicine. We say in the business, when you know one academic medical center, you know one, and that's because the money flows differently at every single one of them. And so, yes, the money flows differently at Washington University and BJC Healthcare. But Jeffrey really taught us all during the time that he was running UPMC of what I call the virtuous cycle of academic medicine. And part of that virtuous cycle, which basically is we take care of patients today and we invest in education and research, and that leads to improvements in the clinical care that we give. And part of what his genius was to figure out where the money is and dedicate that money to the university. And I reflect frequently on the whole idea that when they started, they named it University of Pittsburgh Medical Center, and for the most part stayed true to the idea that the health system was providing resources for the education and research that went on at the university. And also how that paradigm had such an impact on all of us during COVID, when really it was research that had gone on in laboratories of academic medicine that helped us so poignantly during the COVID pandemic. I want to just say that I recently had the experience of spending a day with Dr. Fauci, who came to WashU to give the commencement lecture for the medical students. And it's actually now about 3 or 4 weeks ago. And we had an amazing day with Dr. Fauci. He comes with 7 federal marshals. They are trailing him every minute of the day. His wife also has a detail of 7 federal marshals. And we got into some conversations. I'm telling you this story for a reason. I asked him, I literally was with him all day long, and I asked him, when did things get really bad where you had to worry about yourself? And I was surprised to find out that it actually occurred in April of 2020, when he was being put out by the White House onto the microphone, but the White House was actually undermining him through social media at that time. And so I said to him, did the communication team at NIH help you? He said, no, the communication team at NIH was consumed with Freedom of Information Act request from Congress for all of his emails. Because I think all of you know that even at that time, early on, there was a lot of activities to try to attribute what went on in China to the NIH and to Dr. Fauci. So, but what was amazing about the day with him is despite all of this, and he did recount to us that there were people who were apprehended with weapons by his detail. Through the day, it was absolutely amazing to see what he's like and how dedicated he is to those things that we had to do during the pandemic. And later at night, we were at dinner, and I asked him, Tony, did you remember the question that Vicki Fraser asked you at the end of your virtual grand rounds that you did in St. Louis in the middle of the pandemic? So he had done a virtual grand rounds at WashU in the middle of the pandemic, and Vicki Fraser, who's the chair of medicine, and Vicki is the one of all of our department heads that is always asking everyone, how are you doing? How are you taking care of yourself? She says, Dr. Fauci, you must be working really, really hard, starting at 6 a.m. in the morning ending at midnight. How are you taking care of yourself? And Fauci actually laughed in response, in a way surprised. It's in hundreds and hundreds of interviews. Nobody has ever asked him that question. And he answered back, he said, well, it's what it's like when you're a resident and your shift is over and you learn that somebody is coming in with a heart attack. Guess what? You got to take care of the patient. And that's the way he lives his life. And it seemed to me just perfectly characteristic of what we did during COVID, how we handled COVID, and what we are like in our profession. We figure out how to get things done when we have to get them done. And this is what we do. I'm sure all of you were doing in Pittsburgh, and this certainly was all of what we were doing in St. Louis. And to me, it's what is really remarkable about the academic medicine paradigm that all of us. So those are my stories. It's really wonderful for me to be back and looking at all of you who I had such a wonderful experience here. Barbara and I don't know if either of us are going to cry throughout the day because of how we feel being back with all of you. And I very much thank you for giving us this opportunity to be back. Okay, so I'm supposed to talk about something important. I thought what I would do is tell you a little bit this morning about two projects going on in the lab. And yes, I've been amazingly able to keep going in the lab. And I will say these two projects have been baking in my head for over 25 years. And in both cases, what's fantastic is they are starting, they're in many ways, they're the end of a phase, but they're the start of a very exciting phase. And so what I'm going to do is do a bit on the background. I'll spend a few minutes talking about the background, and then I'll describe to you the two projects and try not to get into too much of the detail. So my interest in the laboratory, well, really, since I started working on alpha-1 antitrypsin deficiency, is described by this picture. These are 3 boys who are homozygous for the variant that causes the classical form of alpha-1 antitrypsin deficiency. The one on the right is actually the middle child, even though he looks smaller. He underwent a liver transplantation at age 18 months. The other 2 are homozygous for the variant and never had any liver disease. These boys are now 25 years old. And remarkably, the index patient has had an almost perfect life after a liver transplant at 18 months of age on a small dose of immunosuppression. And the other 2 have never had a problem with their liver disease. And so the question that has obsessed me is, what is the difference? What is the reason that the one boy had liver disease? And it gets at what's the pathogenesis. It gets at the whole personalized medicine paradigm. And it will, as you will see in a moment, gets at what do genetic modifiers and environmental modifiers do to the pathogenesis of disease. So I'll just remind, many of you heard me talk about this in the past. Alpha-1 antitrypsin is predominantly synthesized in the liver, is secreted into the bloodstream, and its predominant function is to inhibit neutrophil proteases. So it travels in the bloodstream and it protects the lining of the lung. In the classical form of alpha-1 antitrypsin deficiency, the variant alpha-1 antitrypsin, which is called alpha-1 antitrypsin Z, is unable to traverse the secretory pathway. It accumulates in the liver itself, so that now there is a lack of anti-neutrophil elastase activity, and neutrophil elastase can destroy the elastic tissue backbone of the lung, and this is the cause of emphysema. This is how this was first described. That emphysema is exclusively at an adult age, and it is exacerbated by cigarette smoking. The one part of the story that was recognized later was that a small group of the affected individuals would get severe liver disease. And it was first recognized in infants, and infants who eventually in the 1980s, when liver transplantation became possible, would get that liver disease. And it was really that experience that I had as a fellow at Boston Children's, taking care of a few children in the New England area who had this disease and who were unable to raise the money for a liver transplant in 1980 and 1981. Going back in memory, liver transplantation had started at Children's Hospital of Pittsburgh by Dr. Starzl for patients with biliary atresia, but it wasn't readily available anywhere else in the country. And the families had to raise $300,000 or more in order to get it done here in Pittsburgh. So there were families in New England that I was taking care of who couldn't get treatment for this. And I actually began in the laboratory at that time, learning about science for the first time, working on a project that related to how a particular protein got secreted. So the project was about the biology of protein secretion. And I remembered these patients, and that's how I started working on the problem. And I remembered that when you look at the liver biopsy of a patient like this, what you see is here in this slide, this is an H&E, but this is a band of fibrosis over here. So this is a fibrotic liver disease, eventually becomes cirrhosis. And then the dark arrow shows you the accumulation of the variant in the liver cells. Those are the PAS positive diastase resistant inclusions that are the histological characteristic of the disease. So what do we know about this disease? And these are the things that we know about the disease even today. There's a single amino acid substitution that alters the folding of the secreted protein and renders the protein prone to aggregation. And the process leads to selective retention of the misfolded antitrypsin Z in the endoplasmic reticulum of liver cells. 3, we know that the liver disease that is caused by this is due to the retention of the misfolded protein, what will be called by geneticists a gain of function mechanism, or by some proteotoxicity. And this is a paradigm of enormous significance to a bunch of different diseases, the most dramatic of which are the neurodegenerative diseases that affect us as we age. And then this fourth point is the really important point that the Perlmutter was obsessed with all these years: marked variability in phenotypic expression of the liver disease. Only a small subgroup, 8 to 10%, maybe up to 20% of the affected homozygotes develop clinical liver disease. This observation led to a concept that we had, which was first that genetic traits unlinked to the antitrypsin gene and/or environmental factors would predispose to liver injury, but more importantly, the prediction that these putative genetic traits and/or environmental factors would affect the fate of the mutant antitrypsin Z molecule within the secretory pathway, or the response of the cell to its retention within that pathway. Another way to say this is, if the deficient individual bears a trait which reduces the efficiency of degradation of the mutant antitrypsin Z once it has accumulated in the ER, or blunts the protective cellular response, that individual is susceptible to liver disease. And the way these predictions actually we think about them when we think about the cells is shown in the conceptual model here, with what we would call a protected individual. And we're looking at a liver cell. And in this protected individual, the variant is accumulating in the endoplasmic reticulum. Here's the secretory pathway that goes through the ER to the Golgi and out of the cell. And that that variant, that accumulated variant, would be subject to intracellular degradation. And the pathways that are known for intracellular degradation are shown here on the left. The proteasomal pathway, autophagy, and then there are a bunch of pathways that are less well described and probably of less dominant role, Golgi to lysosome. There probably are other pathways. These are the known pathways for this to be graded. One of the big breakthroughs in the 1990s was the realization that a protein that accumulated in the ER could be degraded in the cytoplasm by the proteasome. That actually has a name. It's called ERAD, ER-associated degradation. And secondly, on the right, that there are signaling pathways that help the cell adapt to the presence of a protein in the wrong location, in the wrong conformation. And here we list these pathways as NF-kappa B, autophagy, TGF-beta, caspases, others. These are ways in which the cells and the tissue tries to adapt to the situation. And the conceptualization here is that there's some genetic modifier or environmental modifier that subtly affects either those degradation pathways or the signaling pathways that help the adaptation. That are the key to which child gets affected or which adult gets affected by this disease. I'm going to return to this conceptual model, but I'm going to point out that autophagy sits on both sides here. We know that autophagy participates in the degradation of antitrypsin C in cell models, number one. We know, too, that the autophagy system is activated when antitrypsin Z accumulates in the ER. In fact, it is sufficient, the accumulation of antitrypsin Z in the ER is sufficient to activate that response. So autophagy sits on both sides of this conceptual model. So what is autophagy? I call it the cell's natural vacuum cleaner. And this is a easy way to describe it to people who are very wealthy and are going to give money. It removes waste and harmful agents, damaged proteins, accumulating lipids, worn out organelles, invading microbes. It occurs in every cell of every organism. It is activated by exercise and caloric restriction, the very things that we associate with resilience and longevity. This has become an explosively important part of our physiology. In 2016, Yoshinori Osumi, who characterized the genes of the autophagy pathway in yeast, won the Nobel Prize for that. And the other most important thing is the autophagy mechanism declines with aging in every organism. That decline is hardwired into every organism. And how does it work? There is an isolation membrane that somehow forms in the cytoplasm. Actually, the formation of this remains a mystery, how that happens. And then it forms a vesicle, and the vesicle matures into a complete vesicle that envelops the material that is destined for degradation. Then it fuses with the lysosome and the constituents are degraded. And very importantly, the constituents are then transported out of the lysosome and play a role in the remodeling of the cell. This is a renewing phenomenon. Autophagy auto-digests. We eat ourselves to renew and rejuvenate. This is how you would imagine, these are just cartoons to help you imagine this process. Here, an autophagosomal vesicle is forming around a protein aggregate in the cytoplasm. This will be what I'll talk about when I come to neurodegenerative diseases. Here's a cartoon of an autophagosome pinching off a protein aggregate within the endoplasmic reticulum. This is probably what goes on. This actually now has a name in the field. It's called autophagy. And actually, this process is responsible for the formation of the ER. But it is also as a homeostatic phenomenon. And here's just a cartoon that shows you really what happens in the cell when this process, here's the vesicle coming, it's got a target that it's going to degrade, and then it docks and fuses with the lysosome. But actually, the lysosome injects acid and proteolytic enzymes into the autophagosome, and then the autophagosome becomes called an autolysosome. And then these constituents are then recycled out of the autolysosome. Wait, how do I get out of this? And so what do we know about this from an aging perspective? That is that this declines with aging, and because of this decline in function, there's accumulation of worn out organelles, toxic proteins and lipids that lead to cellular damage. And here you're just given a picture of the atherosclerotic plaques and the plaques and tangles that are known to characterize Alzheimer's disease. We know that all of these aging phenotypes are reversible in experimental animal model systems in which you manipulate the autophagy pathway. So if you reconstitute the autophagy pathway with aging, either genetically or pharmacologically in an experimental animal, you will have an impact on all of these aging phenotypes. So I want to go back to the conceptual model and tell you about the first project. And I'll go back to, as you look at this conceptual model, the first question is it true that in a susceptible person, antitrypsin Z is not degraded as well as in a protected person? And this was published actually in 2015, the year I left, in collaboration with Ira Fox and Edgar Tafeling in Ira's lab, doing a study in which we asked the question, do differences in rate of intracellular degradation of ATZ correlate with the liver disease phenotype? And this was done by doing pulse chase radiolabeling to determine the rate of ATZ degradation in IPS-derived hepatocytes from ZZ individuals with or without severe liver disease. Ira had developed the ability to make well-differentiated hepatocytes from IPS cells. And so we did this in a series of alpha-1 antitrypsin deficient patients who we knew who either had liver disease or did not have liver disease. And the post-chase approach allows you to look at the rate of disappearance of the molecule from the cell. And here you're shown on the vertical axis, the percent of antitrypsin Z that remains. And then on the horizontal axis, the time of the chase period. And you can see in the red are the cohort with severe liver disease. In the blue are the cohort without liver disease. And there is a clear difference in the rate of degradation. It is better in those who are protected. And I'm going to use the term protected as the term for those people who do not have liver disease, but have the ZZ genotype. So that established the idea, yes, that the first part of that conceptual model is right, in as much as that cohort of patients that we examined. So the next question we asked, and this was with Lone Murphy and Beat Neifeller from Novartis, and Gary Silverman, who was my collaborator here and again in St. Louis. Ira and his lab did the analysis of the IPS cells. And Peter Jin is the geneticist who worked with us on the analysis of the genomic sequencing. So the question we asked, and we started this here in 2014, but it has continued until the present. Can we identify the genetic modifiers? And the basic approach here was whole exome sequence analysis using the unique family that I showed you in the first slide and small cohorts of patients that were well characterized. And then the testing of any variants that were identified in either mammalian cell line models or ultimately in IPS-derived hepatocytes from the patients. And so this started with genomic sequence analysis of the 3 boys that I showed you in the first PowerPoint. And you'll remember that the one boy is ZZ and had a liver transplant at 18 months. And the 2 other boys have been absolutely fine to age 25. And we have IPS-derived hepatocytes from each of them. And we could go on to look at an analysis of the kinetics of ATZ secretion in those cells after identifying potential genetic modifiers using exome sequencing. So the first thing we did to validate that this approach would work was actually take those 3 children's iPS-derived hepatocytes and ask the question, does the index patient have a slower rate of degradation than his 2 brothers? And the answer is yes. The index patient here is in red. And if you just look on your right, you're seeing the time of the chase period, and his ability to degrade the ATZ is slower than his 2 brothers. So the idea that validated the model applies to this family. So now we went and analyzed the genomic sequence on the 3 boys and his parents. And the parents were indeed heterozygotes each. And we analyzed a bunch of variants. We used 2 filters to generate the likely genes, and a bunch of assumptions were made at this point. So 113 genes with what were 115 potential damaging variants, 10 recessive, 105 dominant. And by that, what's meant, when we used the word recessive and dominant, what was meant in that case was that the variant is present in the index case as a heterozygote and not present in either one of his brothers. So here, when you get to this point in a project, the idea here, because they're genetically related, is you're improving your signal to noise because they're genetically related, but you still have an issue of having lots of different variants to figure out which ones are pathogenic or not. And that's the holy grail of this project. The other assumption that was made here was that we would look at what would be called damaging variants. So these are variants in exons. So these are coding regions. So we're not looking at non-coding regions. And the variant is predicted to cause damage to the protein function. And lastly, we ended up focusing on variants that were in genes that made sense based on the theory. And so here's a list of a smaller list from the 119 that we generated. This is all the human robot that looks at it and generates this list. And then 2 caught our eye. A gene NTMR12, myotubular related 12. And this gene is known to play a role in autophagosome lysosome fusion. And there actually is a variant in this gene in a known rare degenerative myopathy. And the other one that caught our eye was FAM134A. This is part of a family of genes, the most famous of which is FAM134B, which is now known in the field as the receptor for phagy. That is, it is the molecule that serves as the starter of an autophagosome that is degrading the ER. So these 2 genes seemed pretty interesting to us. We decided to validate them. And so the first thing that was done to look at this question was to see what happens when you use CRISPR to correct the variant in NTMR12 in iPS-derived hepatocytes from the index subject. And you're shown, again, that same format of the data where the amount of the ATZ present is on the vertical, the hours of the chase is shown on the horizontal, and the index patient is shown in red. The protected SIB is shown in black, and then several of the clones done on several different occasions. And what this shows you is that the correction of that variant changes the kinetics. So that looks like a good variant. Next, we did the FAM134A, and yes, this variant corrected the slowed degradation problem. So now we want to go even more rigorous. We're going to introduce the putative pathogenic variant into the protected SIB. And if it really is the right gene as a modifier, it should slow down the kinetics of degradation in the protected SIB. And so here was the result when we introduced this NTMR12 variant in the high heps from the protected SIB. And without spending a lot of time here, you can see it didn't do the job. And then we went and looked at FAM134A. And again, it did not do the job. It didn't change the kinetics in the protected SIB. This data, which we just got a few months ago, is what happens when you introduce both the NTMR12 variant and the FAM134 variant into IHEPs from the protected cells. You now change the kinetics, and it has now become slowed down. And this then qualifies these two variants as genetic modifiers of the liver disease of antitrypsin deficiency. And so in conclusion, susceptibility to liver disease in a subgroup of antitrypsin patients involves genetic and environmental modifiers, and we actually have proven that definitively in this project. 2, IHEPs model personalized variations in liver disease phenotype. This is really a remarkable story, and I glossed over it. We demonstrated this in using fibroblasts from these boys when they were 1 year of age, and then we had fibroblasts from them when they were in their 20s. And we could see that this difference in degradation was hardwired from the time they were born, which means to us it was genetic. Exome sequencing of that family, and I haven't had time to show you, we've gone on to several cohorts. One cohort of 30 patients, and it's somewhat embarrassing to me that there isn't more cohorts that have been well characterized in this disease. There is one cohort of 30 patients who underwent liver transplantation in which you can validate that all of the patients were ZZ. So we did look at that cohort, and then we looked at a cohort of 150 ZZ patients who have neither a liver nor lung disease, and they're in their adult years. And those were sequenced for us to be sure that these variants are truly fit the category of being modifiers. And they have a very low minor allele frequency in the control population. But what we found is that these are rare. And so it's like the autism story. These families are going to have variants that are different from one family to another. We found one other patient in that cohort of 30 who had the NTMR12, the exact NTMR12 variant. We haven't found the FAM134A in any of the rest of the cohort. And if you look at all of the potential variants in the cohort of 30, you do not see a dominant variant. So here I say exome sequencing in a unique family and cohort with severe liver disease identifies multiple private variants in proteostasis genes as candidate modifiers. And then lastly, using the genome editing in IHEPs from affected subjects here that I just showed you, variants in the NTMR12 and FAM134A, when together, fulfill stringent kinetic criteria for genetic modifiers of this liver disease. And because they function in autophagy, this project clearly completes the cycle of the hypothesis that Perlmutter has had for years, that autophagy really is a pathway that is important to the pathogenesis of a disease that is caused by an aggregated protein. And I'm going to come back to that point later as I talk about Project 2. So Project 2 really is a project about, can we develop better therapies? You would think that we now know a really important target, the autophagy pathway, for therapies for this disease. And here I'm just saying there really aren't good therapies for this disease other than liver transplantation. Protein replacement therapy for lung disease probably doesn't really change the lung disease after these many years, lung transplantation. So we, and this is work with Gary Silverman and Stephen Pack that began here in Pittsburgh, where Gary and Stephen asked, could C. elegans be used to model this disease and allow us to develop high throughput methods for drug discovery? They created an amazing model and then showed that it really recapitulated what we were thinking it needed to recapitulate. And then they developed a high throughput screening platform. This is a picture of the worms, the ATZ worms, C. elegans, with these, you can see the gene that's inserted in the worm is designed to be expressed in intestinal cells, which are the metabolic cells of the worm. And it has a GFP tag. And so you see these great big green fluorescent accumulations. That then allows an array scanner to see if the worms are in a well that has a particular drug, if it decreases the amount of the green fluorescent accumulation. And that's how you get a candidate drug. Here is the system that was used. It's a 384 well plate. It's put on an automated array scanner. And 1 hour later, you've evaluated 384 drugs. And it shows you a drug on the left. It shows you a room on the left and a room on the right where the drug or the gene modification has had an impact. So you can see what the array scanner sees. And this then was done, with drug libraries, but also with RNAi libraries. And the idea behind the RNAi libraries were we knock out a gene and it has an impact. Or can we go in with software analysis to find out whether there's a drug that's known to affect that gene? So that's called drug-gene interaction analysis. And the drug I'm going to talk about for the rest of my time here came from such a RNAi screen, not from a drug library screen. So this is what was done here was to use a RNAi library that contains 16,000 RNAis. The worm has probably about 18,000 plus genes, so it's almost the complete genome of the worm. And then when we see a hit, that is a gene that changes the accumulation in one of the wells of the worms, then that gene is analyzed using an algorithm for drug-gene interactions. And the first result of this that struck us was glibenclamide, gliburide, FDA-approved drug for type 2 diabetes, works by increasing insulin secretion through action on the sulfonylurea receptor, SUR1. This made absolutely no sense. There was no way to understand how this could be. And this was very early on. And we had to know, was the whole screening idea even valid? So we took gliburide and we examined it in a mammalian cell line model, and to our surprise, it actually reduced the accumulation of ATZ. So we then said glibenclamide has some kind of degrading principle. And eventually what helped us in this was to deconvolute the gliburide molecule. And if you look on all the way on your left, that's the chemical structure of gliburide. And the sulfonylurea binding region is at the top of the molecule. And so we then analyzed analogs that did not have the sulfonylurea receptor binding domain and discovered, and that's G2, and discovered that the G2 has the autophagy enhancing property. So, and that G2 molecule that you're showing here is the minimal drug needed to have the autophagy enhancer property. We've now gone on to do over 1,000 chemical modifications of that G2 using medicinal chemistry. And we were looking for how do we get to a drug-like molecule that could be used, and in particular with oral administration. Well, here's just some of the data. I won't have time to go through all the data. Here's an analog G2-115. And on the left, you're looking at a Western blot. The G2-115 is incubated with a HeLa cell line that has inducible expression of ATZ. And you're just looking at the steady state level, and you can see there's a dose-dependent reduction in the accumulation. And then on the right is a high throughput assay in which you can look at many more doses, and you see the classical drug effect curve. And the IC50 is somewhere in the neighborhood of 100 nanomolar. So this is an active drug, and it works in two different assay systems. The assay shown on the right is actually from a CRO. It's not from my lab. And that, by the way, is another important part of the commercialization process. You want to be able to have your biological phenomenon independently confirmed by a commercial research organization. This was an important part of the validation. Here, we're going to ask the question, does this drug work through autophagy? This is a way to validate that idea. And here, what we're doing, we're taking a G2 analog and we're incubating it with the parent cell line, which is called HTOZ. It's a HeLa cell line with inducible expression of antitrypsin Z. And on the right side, it's the same cell line, which has been genomically engineered to knock out a key autophagy gene. And that key autophagy gene, in this case, is something called XTX17. It plays a major role in the fusion of autophagosomes with lysosomes. And what you can see is that the drug reduces ATZ in the parent, and you see that both in the top and the bottom panel. When the STX17 is knocked out, there's a much greater accumulation of ATZ because it's being degraded by autophagy, and the drug does not work. So this allows us to draw the conclusion that the drug, even though at this point we do not know the molecular target of the drug, that the drug is working through an autophagy-dependent mechanism. So the last few slides I'm going to show you are really the most exciting part of this project. The G2 analog series reduces inclusion bodies and cell death in a human neuronal model of Huntington's disease. So here's the question. If this is a drug that is affecting a key disposal pathway, and it does that in a generic way, it should stimulate the degradation of other aggregated proteins. Huntington is a very, very different molecule from antitrypsin Z, but it aggregates, and it aggregates in a particular part of the brain to produce Huntington's disease. And I think all of you know, a terrible, terrible neurodegenerative disease. What's amazing about this study, which was done by Andrew Yu, a developmental biologist at WashU. Andrew is an expert at cellular reprogramming for brain cells, and he created the most powerful model of human Huntington's disease by reprogramming fibroblasts. This is not IPS, this is reprogramming. Reprogramming is the future of medical research. Reprogramming is a method where you put the fibroblasts into a cocktail of transcription factors and, most importantly, microRNAs that allow you to differentiate the cells in a positive way without any dedifferentiation. And you can make specific subtypes of cells. So in this case, he made what are called striatal neurons. The striatum is the target of Huntington's disease. And because he could make the striatal subpopulation in this system, the Huntington causes neurodegeneration, and it causes the neurodegeneration in the model system in an age-dependent way. And what do I mean by that? Huntington's is dominant. So there are people who have the Huntington's genotype before they ever get the symptoms, the neurodegenerative symptoms. So he can go in and test fibroblasts from people with the Huntington's genotype before they have any symptoms. And what he shows is the cells don't die. When they have symptoms, the cells die. And by the way, he's done phenomenal genomic analysis of that. And what he finds, and there's a paper actually right now, which is going to be accepted in Nature Aging, that what happens in that process is the autophagy system declines. The autophagy system, when he reconstitutes it, they don't degenerate. And here he shows that the G2 analog G2-115 reduces the accumulation of inclusion bodies and reduces cell death in this model system. And then the last data I'll show you is data, it's a little fuzzy because they'd actually, my office took this from my iPhone, and this comes from Celeste Karch, who's a faculty member in the Department of Psychiatry at WashU. Celeste has done all kinds of iPS cells from different patients. And so here she is, she's looking at an iPS-derived cell line from wild type on the top, and then from the patient that has frontotemporal dementia. Frontotemporal dementia is one of the classic tauopathies. And I think all of you know that in Alzheimer's disease, you get amyloid and tau. And so we're interested in frontotemporal dementia, but we're really interested in Alzheimer's disease. And so the question here is, does the drug work on tau in a neuronal model? And so these are iPS-derived neurons, and wild type on the top. The identification of the cells is with green. The identification of the tau is in red. And what you see here is that the G2 molecule reduces the tau accumulation almost to the level of the wild type. It also reduces it in the wild type cells. And here's the quantification over here. So this is exciting data. And I don't have time to show you this, but recently Celeste showed me an experiment in iPS cells from a Parkinson's patient and showed that the drug also reduces the accumulation of alpha-synuclein. So in conclusion, this lead candidate, which is now been modified to the point where it has a half-life of about 12 hours after oral administration and penetrates the blood-brain barrier, is capable of reducing the accumulation of different aggregated proteins. It fulfills the criteria for a drug that could potentially make us live for a longer time. And we're very, very excited about being able to test this now in preclinical models. And we also need to identify what's the target of the drug action. I look like I'm out of time. We do know we are as far upstream in understanding the drug in the neuronal model, and so we're hopeful that we're going to be able to identify the molecular target, because that's a key part of moving this into translation. So I want to thank my lab, which is shown here, and the amazing thing about this group is that some of this group moved from St. Louis to Pittsburgh in 2001, and moved from Pittsburgh to St. Louis in 2015. This is Gary Silverman and Stephen Pack, who made the C. elegans model, and all of you know Gary. And David Rudnick, who was one of my former mentees when I was in St. Louis, is now helping me run my lab in St. Louis. Here's Ira and Edgar, who are here in the building. I'm a little surprised they're not here today, who have done all the work with the iPS-derived hepatocytes. Andrew Yu, who is our reprogrammer, Celeste Arch, who does the IPS cells, and a number of other laboratories that had worked with us, including Yvette Bahar's lab, who did the computational pharmacology, and Rick Wilson, Tom Nicholas, Dan Cobalt, Adam Locke, and Peter Jin, who helped us with the genomic analysis, Andrew Wilson and Darrell Cotton with the making of the iPS cell. So I'm going to stop there, Terry, and I guess there are a couple minutes left for questions. Thank you all for listening.